If you could eliminate disease, boost intelligence, or even choose your child's hair color before birth, would you? The power to edit the human genome is here, but with it comes an ethical tightrope. Are we creating a new, natural selection, where wealth dictates who gets to evolve? And what does that mean for the very definition of being human? CRISPR babies are no longer a sci-fi concept, but a reality. Are we on the cusp of an evolutionary leap, or a dangerous slide into a new form of eugenics? This isn't just about curing diseases. It's about fundamentally altering our species and asking if we're playing God with our own destiny. Before we dive into CRISPR babies, you need to understand what CRISPR actually is. CRISPR stands for Clustered Regularly Interspaced Short Palindromic Repeats. It's a naturally occurring defense system found in bacteria that helps them fight off viruses. Scientists discovered they could hijack this bacterial immune system and turn it into the most powerful gene editing tool ever created. Think of CRISPR-Cas9 as molecular scissors with a GPS system. Here's the three-step process. Step 1. Find. Scientists program a guide RNA, a genetic homing device that searches through billions of DNA letters to find one specific target gene. It's like using Control F to search a document with 3 billion words. Step 2. Cut. Once the guide RNA finds its target, the Cas9 protein, the molecular scissors, makes a precise cut in the DNA strand at that exact location. Step 3. Edit. After the cut, one of 3 things can happen. Delete. The cell repairs the break, but removes the gene, disabling it. Correct? Scientists insert a healthy copy of the gene to replace a diseased one. Add. Entirely new genetic code can be inserted. The cell's natural repair machinery does most of the work. CRISPR just shows it where to operate. What makes this revolutionary? It's precise, relatively cheap, and fast. What used to take years and millions of dollars can now be done in weeks for a fraction of the cost. CRISPR babies are children born after their DNA was intentionally edited while they were still embryos. The editing happens at the earliest stage of life, when an embryo consists of just a few cells. Originally, CRISPR was developed to treat genetic diseases, study DNA, and modify plants and animals. But when applied to human embryos, the changes become heritable, meaning they can be passed down to future generations. This is not therapy for one person. This is editing the human lineage. In 2018, the world was stunned by an announcement from Chinese scientist He Jiankui. He revealed that he had genetically edited 3 human embryos, resulting in the birth of 3 babies, including twin girls known publicly as Lulu and Nana. What did he change? He disabled a gene called CCR5, which is involved in HIV entering human cells. His goal was to make the children resistant to HIV. But here's the problem. The fathers were HIV positive, but there were already safe medical ways to prevent transmission. CCR5 also affects brain development and immune function. The edits were imprecise, creating unknown mutations. Some cells in the baby's bodies had the edit, while others didn't, a condition called mosaicism. This was the first known case of germline gene editing in humans, changes that would be passed to their children and grandchildren forever. The global scientific community reacted with outrage. He Jiankui was fired from his university, condemned by scientists worldwide, convicted in China for illegal medical practices, sentenced to 3 years in prison, fined and banned from reproductive research. And the children? Their identities are protected, but what we know is unsettling. They are under lifelong medical surveillance. Scientists do not know if the edits will cause health issues later in life. Some studies suggest CCR5 deletion may affect cognition and vulnerability to other diseases. They may face social stigma and psychological burden from being the world's first edited humans, and probably prohibited of passing on their genetics to future generations. These children became the first humans in history to live with irreversible experimental genetic changes without their consent. This is where science collides with philosophy. Consent. Embryos cannot consent to permanent changes that affect not just their lives, but future generations. Should parents have the right to make irreversible genetic decisions for children who don't exist yet? Inequality. If gene editing becomes safe and legal, who will afford it? Wealthy families could eliminate disease, enhance intelligence, height, or strength, and create a genetic elite. This raises fears of biological class division, a world where your DNA determines your social status. Slippery slope to eugenics. History has shown us where improving humans can lead. What starts as disease prevention could become cosmetic traits, behavioral selection, and cultural bias encoded in DNA. Who decides what traits are desirable? Playing God. Once humanity edits itself, evolution is no longer natural. We become both the designer and the experiment. Are we wise enough to wield this power responsibly? Unintended consequences. Genes don't work in isolation. Changing one gene can affect dozens of biological processes we don't fully understand yet. What if the cure is worse than the disease? Here's the current global landscape. China. Research continues under stricter regulations post-2018, but embryo editing for reproduction is officially banned. United States. Germline editing for babies is illegal, but CRISPR is widely used in medical research and therapies for existing diseases. United Kingdom. Allows gene editing on embryos for research only, not implantation or birth. Japan. Permits embryo editing for research purposes. Reproductive use remains prohibited. Russia. Has expressed interest in the technology, but officially bans clinical use. No country currently allows CRISPR babies legally, but research is accelerating, and the technology is becoming more accessible every year. Scientifically speaking, no, not yet. The experiment did not cure a necessary condition, prove long-term safety, meet ethical or medical standards, achieve consistent edits across all cells. Instead, it revealed how unpredictable gene editing can be in human embryos, how one genetic change can affect multiple biological systems, how unprepared humanity is for germline control, the critical need for international oversight and ethical frameworks. But CRISPR itself is not a failure. CRISPR has shown incredible success in treating existing patients with sickle cell disease, certain cancers, and rare genetic disorders. The difference? Those treatments edit cells in living patients, not embryos destined to become children. Editing embryos intended for birth remains unsafe, unethical, and scientifically unresolved. CRISPR gives us godlike power, but not godlike wisdom. The question is no longer, can we edit humanity? It's whether we should. Every technology is a mirror. It reflects who we are and who we want to become. CRISPR will force humanity to answer, what does it mean to be human? And who gets to decide? Where would you draw the line? Do you believe CRISPR babies are the future of medicine or a dangerous step too far? Comment below with your thoughts. Subscribe for more science stories that shape our future.